Good morning students. I hope you are watching the video completing your homework. We are studying about the plant tissue. I hope you have learned about the meristematic tissue and simple permanent tissue which I have taught you in the last videos. So we are studying about the plant tissue. Plant tissue is of two types, that is meristematic tissue and permanent tissue. Meristematic tissue means those tissues which are having the capacity to divide, whereas permanent tissue means those tissue which has lost the power of division. So before we start the new topic, let's recall our memory. We are studying about the permanent tissue. Permanent tissue means those tissues which has lost the power of division. Now permanent tissue is of two types, that is simple permanent tissue and complex permanent tissue. Simple permanent tissue means those tissues which are made up of similar type of cells, whereas complex permanent tissue means those tissues which are made up of different type of cells but they coordinate with each other to perform a particular function. Now simple permanent tissue, they are those tissues in which the permanent cells are similar in structure, origin and function. Simple permanent tissues are of three types, that is parenchyma, collenchyma and sclerenchyma. So already we have done this parenchyma, collenchyma and sclerenchyma. Let's recall our memory. Parenchyma means simple permanent tissue in which in between the two cells, see here, the intercellular space is present. See, the intercellular space is present in between the two cells, and this helps to store the food. This is the transverse section and this is the longitudinal section. Next is collenchyma. So collenchyma means, see here, the deposition of pectin is there. This is the deposition of pectin. So due to the deposition of pectin, the intercellular space become smaller and it helps in easy bending of the plant. But don't get confused between collenchyma and chlorenchyma. Chlorenchyma is the type of parenchyma in which the chlorophyll is present and it helps in photosynthesis. Among this also chlorenchyma, you have studied in parenchyma about the arenchyma. Arenchyma means those tissues that contain the air spaces in between the cells that helps to float the aquatic plant in water. So that is arenchyma. So don't get confused between the collenchyma and chlorenchyma. Okay. Next is sclerenchyma. The main difference between parenchyma, collenchyma and sclerenchyma is that parenchyma and collenchyma contain the living cells, whereas sclerenchyma contain dead cell. And here the deposition of lignin is there. See here, this deposition of lignin is there. And this is the cementing object, so it make it hard. And due to this deposition, this lumen become very narrow. And it is present in the covering of the nuts or in the husk of coconut. Now we start the today topic, that is the activity 6.3. Now activity 6.3. So aim of the activity is to prepare a temporary mount of a leaf, that is Rio leaf. Now materials required: fresh leaf of a Rio, slide, watch glass, forceps, glycerine, brush, pair of sharp scissors, safranin solution, blotting paper, cover slip, compound microscope. Now procedure. Take a fresh plucked Rio leaf, break the leaf from front to back by putting a suitable pressure so that the peel of lower surface comes out. Be sure that you are taking epidermis from lower side as the dorsal surface is rich in stomata. Cut small pieces of the peel into watch glasses filled with water. Add a few drops of safranin into the watch glass to stain the leaf peel. After 2 minutes, place the stained peel into water in the watch glass again to wash off extra stain. Now put a drop of glycerin on the slide. With the help of a brush, transfer a piece of peel on the slide. Now put the cover slip over the peel gently by first making an angle of 45 degrees with the slide with the help of forceps. Be careful that the peel should never be folded. Place the cover slip on the slide gently and gradually to avoid the entry of air bubble. Drain out the extra stain by plotting paper. Now examine under the low power of compound microscope. Observation. The cells are closely packed together with distinct cell wall. Each stomata is guarded by a pair of bean-shaped cells called guard cell. Guard cells are rich in chloroplast. So this was the activity 6.3. I hope you have understood. Now write down this activity in your copy as homework and learn for the test. I will announce the test in the next video. So be prepared for the test. I'll take the test most probably on the next week. Thank you.